In dehydration synthesis, we've been focusing in on carbohydrates, and that's this group here, right? So we've been talking about taking simple sugars, for example, glucose, and we said glucose was the most important one to get to know. That this glucose molecule we've been taking and putting together into first disaccharides and then oligosaccharides and then finally complicated polysaccharides. And one of the properties of carbon that we discussed is that carbon gets to branch. So it doesn't have to go in a straight chain, it can branch. So all kinds of shapes and sizes can occur. Because they're virtually unlimited, these carbohydrate chains can be hundreds, if not even thousands of monomers long. Now this word monomer keeps coming up as kind of a confusing point. And I wanted to show you this slide to try to emphasize the idea that a monomer is a general word for any one of the groups that can be linked together in a chain. So here we have a bunch of monomers connected together. We also have that going on in proteins and in nucleic acids. The monomer for polysaccharide sugars, we're talking about simple sugars, things like glucose, fructose, and galactose. We're focusing in on glucose. In amino acids, which we'll cover next week, it's really in proteins, the monomer is amino acids, and these amino acids take up one after the other after the other. In DNA and RNA, it's nucleic acids. So you have that ribose base that we talked about, and then you have some phosphate connected to it. And there'll be some base. That's this B is another variable that represents a fairly complicated structure that's either a G, a T, a C, or A. But these we'll cover in a couple weeks. These we'll cover next week. We're focusing in on these, but the point is that each of these monomers, each of these are monomers. This is a monomer, this is a monomer, and this is a monomer. So how do we distinguish the three monomers? Well, this is a monosaccharide. That's the easiest one to remember because it's mono means it's one of the many and then a chain. Amino acids are just called amino acids. They're the monomer of proteins, but they're called amino acids. The monomer of polysaccharides are monosaccharides. And the monomer of DNA and RNA, or RNA, are nucleotides. So these two words are not the same as the monomer for polysaccharides. But the key is that they're still monomers. So the general term monomer can apply to nucleotides, to amino acids, or to simple sugars. It turns out lipids don't really form polymers. We'll talk more about that later. But these three definitely form polymers, and they deal with dehydration synthesis and hydrolysis. So we need to understand that process. So hydrolysis and dehydration synthesis are just the reverse reaction of each other. And so what you have to understand is that when we have these two monomers, again, monomers, here is the monomer of this disaccharide. This is a disaccharide. This is one monomer of that disaccharide. This is another monomer of that disaccharide. So the question is, what's happening here? You know, a lot of you are having trouble with the notation and the pictures. Some of you are forgetting that each one of these corners of the molecule are carbon. That hydrogen is generally not shown. Sometimes it is, but most of the time they don't show it. So that these carbons always have 4 bonds. So if this is an oxygen here, this should be an oxygen. It is. It's just they drew it outside the circle. Usually the oxygen is in the middle of the bond. It's not standing outside it. But this here is a carbon, and there are 4 bonds to it. It's bonded to this carbon here. It's bonded to this there's another hydrogen down there it's bonded to. And it's bonded to this carbon, and it's bonded to this oxygen. This carbon then therefore has 4 bonds. This carbon here has 4 bonds as well. One bond to this hydrogen, which is not shown, and a bond to an oxygen, and a bond to another carbon. So that's 1, 2, 3, 4 things it's bonded to. So everything here has a complete, they're full. The number of bonds they can make are full. Each hydrogen can only make one bond. For instance, this hydrogen is bonded to this oxygen. This oxygen is bonded to this carbon, and oxygen can only make, that's right, 2 bonds. So every bond is full, so we have to clear off a bond if we want to connect this molecule to this molecule. Remember, these are all atoms. They're all bonded together into some kind of structure. We call these structures molecules. Now we're going to try to connect this molecule to this molecule. How can we possibly do it? Well, the way we do it is we have to kick something out. Something has to be kicked out, like musical chairs, as we discussed in class. We're not going to allow 2 people to sit in one chair. That's a solution, but that's not a viable solution because we're not going to allow it. So nature does not allow more than 4 bonds to carbon, and for that bond and for that situation to be stable. So you can think of it as 3 or 4 people trying to sit, or 2 people trying to sit in a chair, and then the chair just breaks. So this is what would happen if you try to make more than 4 bonds per carbon. There would just be an issue energetically, and it would just fall apart. So what happens is that this carbon here, in this position carbon 1, has 4 bonds, 1 to this hydrogen, 1 to this oxygen, and 1 to this carbon, and then the 4th bond being to this oxygen. This one clears that bond that it has with the hydroxide by taking hydroxide and having it come out. Now that takes care of one of the bonds that need to be cleared, because if you're trying to bond it to this one also needs a bond cleared. So rather than kicking this hydroxide out as well and then having 2 hydroxides out here, it turns out that this hydrogen here leaves, and that allows for this oxygen here to bond to that carbon. So you end up with H2O out here, right? That's the hydroxide from this, and the hydrogen from this. And then this oxygen bonds to this carbon. Remember, this carbon used to have 4, now only has 3. This oxygen used to have 2, now only has 1. It's still just bonded to this carbon. This hydrogen is not there anymore, it's out here. So then that oxygen can now make a bond between this carbon and this carbon. So these 2 groups of atoms that are bonded together are now all one group, and we call that group a disaccharide. And I always spell disaccharide wrong because I keep forgetting to add the 2 C's, but a disaccharide is di for 2, and then saccharide means sugar. All right, so then you have a disaccharide is just 2 sugars bonded together at this glycosidic bond. Now, you don't need to know it's a glycosidic bond. All you really need to understand is this process where this hydroxide comes out, this hydrogen. Now you might ask, well, if these were stable molecules, how come this happened? Well, remember, there's one thing we talked about repeatedly, actually, and we even did a little a lab on it really quickly, is this idea that we haven't really looked into too closely yet, but we will, is this idea of enzymes. And enzymes run this. The reaction will not happen unless you have an enzyme that comes in and helps it and does it. And it requires a specific shape, and because of that specific shape, because enzymes do what they do, it allows water, H2O, to come out, and the 2 monosaccharides become a disaccharide. So now when we're talking about the reverse reaction, as you see here, the direction of the arrow that you see here is showing you that we're moving in this direction. So you're taking 2 and becoming 1. Again, we call that dehydration synthesis. That's pretty clear. Now, when we're doing the opposite, it's just running this reaction backwards. Now, we can't do that reaction. We can't take this disaccharide and break it up, or it could be a polysaccharide. It doesn't have to be a disaccharide. Every time you put one of these together, you get a water out, and the chain gets longer and longer. But no matter what, if we run in the reverse reaction, we need a new enzyme. So this enzyme is going to do something called hydrolysis, right? Hydrolysis, and we talked about this. It means breaking up using water. So we're going to go ahead and do that, and we're going to reverse this reaction. Remember, a lot of reactions and it can run backwards. We can make the reaction go in the opposite direction. Just because a reaction normally goes in, this direction doesn't mean we has to go always in that direction. We can, under different conditions with a different enzyme, we can make the reaction go in the opposite direction. There's a really good video on YouTube if you'd like to go watch and find it. And some scientists have recently been able to reverse the chemical reaction and makes a fried egg. When you cook an egg, anytime you cook, you're doing chemical reactions. And it turns out that we used to think, once you fry an egg, it's done. You can't change, you can't go back. But it turns out if you fry an egg under certain conditions with specific enzymes, you can make the egg go back to being unfried. We've been able to unfry eggs. That's how cool these enzymes can be. We can actually reverse the reaction that made on a fried egg. So we can make reactions go in opposite directions. In fact, often they go in reverse on their own. It's just a matter of degree as far as what we see with our eyes. If we have a few molecules going in this direction and many more going in this direction, what we see with our eyes is the breakup of the disaccharide. But if we have just a few of them breaking up, but then a lot of them being made, what we see is the making of the disaccharide, because that's the average, is what we call equilibrium, and we'll be talking about this later. But reactions often go in both directions. In fact, this single arrow in one direction is rarely the case. We can usually, these words usually and rarely, often we can take that reaction, and most reactions are going to go in both directions. It's just a matter of in which direction is it happening more, and that's what we see, right? The stuff that we don't see is the stuff that's overwhelmed by the majority. So let's go ahead and take a look at the next, at hydrolysis, and then which is the reverse of this dehydration synthesis. But let's look at hydrolysis in detail, and then we're done with this short video. When we're taking a look at hydrolysis, it's again really the opposite of dehydration synthesis. I hope that you guys can see the difference between this and what we started off with when we looked at dehydration synthesis. In dehydration synthesis, water was on this side of the reaction equation. In hydrolysis, water's on this side. That's because we're using water and we end up with no water over here. So we use the water to break these up. So let's go ahead and try to figure out what's going on here. When we're looking at water, you got to remember that water is in fact an oxygen bonded to two hydrogens. And when we're remembering that each of these carbons, this is an oxygen, each of these carbons at these points, they have 4 bonds. This has 4 bonds, this has 4 bonds. We're really focusing on this one and this one, these 2 carbons, and this oxygen when we're talking about trying to break this up. This is the bond that's holding them together, and we have to try to break them up. What ends up doing is we just do the opposite of what we did to make this. And what we do is one of these hydrogens is going to end up going with this, is going to end up going into a bond, making a bond with this oxygen, and it ends up over here as a hydroxide. Then this hydroxide here ends up going and filling in the missing bond that carbon needs now that you've broken the bond between these two. Now that you've broken this bond, this carbon only has 3. Remember, in nature that's not acceptable. So you have this hydroxide that comes in and bonds to this carbon, making this hydroxide. making this hydroxide here. So this hydroxide came from water, where this one over here only the hydrogen came from water. You'll notice the color coding. These two are red, this is red, these two are red, and this hydrogen is red. The oxygen is black because it was here originally and it's still here. All right, so every oxygen has 2 bonds, every carbon has 4, every hydrogen has 1. We are a go on both of these molecules, and so what you have now is 2 glucose molecules, and you started with over here a disaccharide, and probably, I'm not sure, but probably maltose, right? Because these look like 2 glucose molecules, and when you have 2 glucose molecules put together, maltose, remember, you don't need to remember those, but it's good to remember that ose is sugar, and that any disaccharide can be separated using this hydrolysis. So we have this hydrolysis and dehydration synthesis. Let's remember that dehydration synthesis, another word for it, is condensation reaction. So when we're talking about condensation reaction, dehydration synthesis, you're talking about putting monomers together, one after the other after the other. And every time you do it, you make water, because these two have to come off. Remember, carbon can only have 4 bonds and oxygen can only have 2. If you're going to make these covalent bonds happen, you have to allow oxygen to have an available bond, and you have to have this carbon to get rid of its hydrogen to have an available bond. So it turns out again that this hydrogen goes off in this direction, this hydroxide goes off in this direction, and they form water. And then the new bond that's formed is between the oxygen that's left over and the carbon that's on this side. So that's dehydration synthesis. Remember that another name for dehydration synthesis is condensation reaction, condensation synthesis. It's the same thing as dehydration synthesis, right? So you need to remember that. And that's just taking a monomer, bonding them together covalently to form a polymer, and taking water out. With hydrolysis, it's the opposite. So you're taking water now, and you see here there's water, and the hydrogen is going off with the oxygen that was bonding the two, and there's the oxygen and the hydrogen from water. That one of the hydrogens is there, and it's forming its bond, it's moving away, and then the hydroxide from the other goes and joins over here. They really should put an OH here, I guess. And that shows that there you have the hydroxide in one group and a hydrogen in the other, and they separate into a monomer. This is a monomer, right? Now this monomer can be anything. This could be a protein we're looking at, or this could be a DNA molecule we're looking at. Or in this case, because we've been focusing on carbohydrates, if this were a carbohydrate, this monomer would be a monosaccharide, right? So it's the idea that, and the reason that we use this word monomer instead of monosaccharide is that this process is the same for all the biomolecules that form these. In fact, this is the same for any polymer. It doesn't have to be a biomolecule, but let's just stick with biomolecules. Of the 3 biomolecules that form polymers, they're all of them have monomers, but only carbohydrates have monosaccharides. So try to remember that the idea, the relationship between the word monomer and monosaccharide, because that seems to be a real confused point among some of the students in the 9th grade this year. All right, well, I think that covers dehydration synthesis and hydrolysis. I hope that helps all of you. Remember all the other videos and all the other lessons in class and the PowerPoints. Don't forget the book. The book is a great source, has their own links to their own videos. You can find your own video. But I warn you, some of the students have been finding, at least one student found a video that actually misled her. She thought she understood, but still didn't. You have to check your knowledge. It's good if the video helps you. Just check your knowledge. Make sure you're answering correctly. Make sure you get the answer correct every time you ask someone asks you what is a monomer, if your understanding leads you to the right answer, then we're ready to go. But if you get it wrong, if you're making you're doing your project and it doesn't seem to be coming out, then you have a misunderstanding you got to fix. I will be sending out the video on the project today. I'll be answering all the questions. All you have to do is fill out the answers on your paper and get credit for it. Last block already finished the lab. They produced their polysaccharide on, and they've stapled it onto the board. I'm hoping the rest of the classes will be able to do theirs tomorrow after the quiz. The polysaccharides that were looking good when I left. I will again explain the project to you. Hopefully this whole thing, if you use this, you should be able to do the project. Those of you that don't have a copy, paper copy of the project, I will be emailing you a version or the copy of it so you can print it at home or at the library. Not sure the library is an option, but at least you don't even have to print it. You can just draw it out by hand using the electronic version as your template. Draw it out by hand and cut it out and do it yourself. You can draw these things by hand. You don't need to have a printed copy. Alright, good luck, guys. There'll be another short video explaining and giving you all the information you need for the project that we started in class. One class completed. Good luck to you.